ImmunoX program. It's my great honor to introduce our distinguished guest today, Dr. Barney Graham. Dr. Graham earned his MD at the University of Kansas in 1979 and completed a medical residency and chief residency at Vanderbilt, where he took care of Nashville's first patient with AIDS. He went on to complete an NIAID fellowship and earn a PhD in microbiology at Vanderbilt, all while leading the first clinical trial of an HIV vaccine. In 2000, he was recruited to the Vaccine Research Center at NIAID, where he has served as its deputy director and the director of the Laboratory of Viral Diseases, where his laboratory has made groundbreaking discoveries facilitating vaccine development for RSV and Zika viruses. These discoveries and his clinical trials work laid the foundation for both the rapid development of highly immunogenic and effective mRNA vaccines for COVID-19, as well as the required regulatory framework to design trials that could deliver approved vaccines in less than 1 year since the initial identification of the virus. This has to be considered one of the most important scientific developments of the century, and Dr. Graham was at the center of it. So in December of 1854, the great vaccinologist Louis Pasteur said that in the fields of observation, chance only favors the mind that is prepared. Well, in December of 2019, it's hard to imagine a mind more prepared than that of Barney Graham. One of my VRC colleagues recounted to me that at a VRC PI meeting in December of 2019, just as news of a cluster of cases of pneumonia was being reported in Wuhan, China, Dr. Graham leaned forward in his chair and said, "Well, I think this is a coronavirus," and the rest is history. So Dr. Graham certainly met the moment, and we are all both grateful for his contributions to science and global health and inspired by his story. We're excited to learn more about his story in a moderated discussion after his prepared remarks. The moderated discussion will be led by Dr. Monica Gandhi, our CFAR director, and Mark Ansel, our ImmunoX program director. And we'll follow that by an open Q&A period where we invite you to ask your own questions. So without further ado, please join me in welcoming Dr. Barney Graham. Thank you for joining us.
Well, thank you, and good morning to you all. Afternoon to people on the East Coast. It's good to see a lot of friends that I've worked with over many years. I didn't know I'd get to see other CFAR people coming on, but it's nice to see you all.
You're very
I'm going to just briefly, let's see. Give you a few disclosures. I'm an inventor on vaccine and antibody patents for a number of viruses. I wanted to start actually back with Louis Pasteur and Jenner and talk about how advances in vaccine development have often come opportunistically based on other technology developments. And so these first two, Jenner's and Pasteur's antiviral vaccines, were done before vaccines or viruses or anything was even understood, before immunology was discovered. And these were grown in animals, either in the spinal cord or on the hide of a calf. These vaccines were grown in animal tissue, living animals. When Goodpasture learned how to grow viruses in eggs, it allowed us to develop influenza and yellow fever vaccines. But the real change came when Enders et al. Developed cell culture technology that allowed really high titer virus growth and allowed a number of live attenuated and whole inactivated viruses to be developed. Things changed again when molecular biology took over and allowed recombinant proteins, virus-like particles, reassortant viruses, molecular clone viruses to all be something that could now be made. And it also evolved to cell culture or production of proteins in bioreactors. And now, recently, we had the VSV gene-based delivery approach approved for Ebola, which is sort of into the new era of vaccinology that I think of as largely focused on structure-based design, but also a lot of other types of technology that have really come up largely in the last 10 to 12 years. And we thought RSV might be the first example of structural biology, structure-based vaccine design, but SARS-CoV-2 came in and basically utilized all the different technologies that I have listed here. And so I just want people to understand that most vaccine advancements happen because of other technologies. And I also don't want to forget, we want to ask, the VRC was founded to develop an HIV vaccine, and I think that's really still at the heart of why all of us that were at the VRC have done this kind of work. And I'm no longer at the VRC, but it's still a high level goal for me to see the field develop an HIV vaccine. And I just think we need to remember that Dr. Sabin, who you shouldn't underestimate, didn't think an HIV vaccine would be possible. And he had his own reasons. I have some of my own ways of thinking about it, and my reasons why I think it may be difficult, hopefully not impossible, to make an HIV vaccine. And part of it just has to do with the type of virus that HIV is. Respiratory viruses and other acute infections often have virus growth curves and then illness curves that look like this. You might get some disease during that initial burst of replication, but a lot of the disease that we see in respiratory viruses comes as virus titers are on the decline. This is why you have to really treat early with things like oseltamivir to have an impact on influenza. And these are the kinds of viruses that traditionally have been susceptible to vaccination. Viruses like this, where the disease really tracks with the virus load, like herpes and HIV, for instance, or HCV, often have not often had vaccines developed. We have a zoster vaccine, but it really is a boost to prevent recurrence and not really something to prevent infection. The other thing I just want to remember is that I made this slide back in the 1990s, but it's still true to a large extent that antibody arm of our immune response is what we use to prevent infection, and T cells are what we need to rapidly clear virus-infected cells. But once latency or sequestration or immunoprivileged sites are inhabited by HIV, neither of these work very well. So whatever happens has to happen relatively quickly, and it probably needs both arms of this immune response. Because I think this is what we hope for in respiratory virus vaccines, is an attenuated, a rapid immune response, attenuated growth, reduced inoculum size, so that we prevent the illness altogether and keep it subclinical. And in HIV, I think the things we can hope for would be shown here, either controlled persistent infection or ideally a rapidly aborted infection. And it's really, in my opinion, a matter of timing. During HIV infection, after it goes through this bottleneck, and then systemic spread inhabits the lymph nodes, and then antibody responses and T cell responses can control it to some extent, but they all come too late. But if you could have pre-existing antibody and a very robust CD8 response, it's possible you could get onto one of those other curves. So these are the outcomes that could happen with an HIV vaccine. One of these two would be ideal. this would be less ideal, and we're trying to avoid disease enhancement, but that clearly is a possibility that has to be dealt with. So this, for me, the coronavirus story really started with HIV, but it went through RSV, and our work on RSV led to capturing this pre-fusion form of the F-glycoprotein. This was done in collaboration with Jason McClellan, who was a postdoc in Peter Kwong's lab at the time, and finding an antibody that can neutralize but not bind the post-fusion form of F, and then holding C-terminus together with the fold-on trimerization domain allowed the capture of this protein, which is otherwise very unstable. And that's then allowed us to map epitopes and understand finally how this post-fusion form of F, which has been used as vaccines for several decades, does have some of the neutralizing epitopes preserved, but they're most of the ones that are less potent. So the more potent neutralizing sites on RSV are all reside at the apex of the pre-fusion form of the molecule before it rearranges. And so just to give you a sense of that, this is the site 0, the most neutralization sensitive epitope on the virus. It was discovered during that process of crystallization, and it is lost when the protein rearranges into the post-fusion. And this molecule, traditionally over at least 5 efficacy trials, gives you only a 2 to 3-fold boost in neutralizing activity, whereas if you stabilize with this C-terminal trimerization domain and internal disulfide and cavity filling mutations, you can now achieve upwards of 10 to 20-fold boost in neutralizing activity, which now becomes a vaccine. And just to be clear about how dramatic this rearrangement is, protein starts on the virus surface and then completely unravels, at least the top unravels, inserts in the host cell membrane, and the heptad repeats come together, cause this membrane fusion so the nucleic capsid can go in and start the infection process. So we knew this happened, but I don't think we really appreciated the consequences of the dramatic nature of this rearrangement up until the last few years. So RSVF is a class 1 fusion protein. It's like other class 1 fusion proteins. For instance, it's very similar to parainfluenza virus. Both components of this protein are kind of wrapped together and they all move together. There are other class 1 fusion proteins that have this cap, like influenza and HIV, the GP120 is the cap. Ebola and Lassa are all have glycoproteins that are class 1, where the cap has to come off for the fusion machinery to act. And then coronavirus spike has all of these components. It has the fusion machinery with this intervening piece and the cap that has to come off before the fusion machinery can act. So they have a lot of functional homology and shared domains and motifs, but obviously very different shapes and topography. But since they work the same, there is learning that can go across these different virus families. And so when Jason McClellan went to Dartmouth at the end of 2013, just as we finished the RSV work, and he was looking for a new area. We were just coming out of the MERS crisis where we had made some vaccines but weren't very happy about them. And so we decided to continue our work together and extend the findings from RSV onto coronavirus. And after 3 years with Andrew Ward's help using cryo-EM, we were finally able to get the structure of the spike of a human coronavirus, of HKU1, one of the endemic coronaviruses. And this is what it looked like. And going through the iterations that we often use to stabilize these proteins, we found the proline substitutions at the top of the central helix of the fusion machinery would be right about in here, inside the protein, largely stabilized this. It prevented the cap from coming off. It's very similar to what John Moore and others did for the SOSA molecule of HIV. And interestingly, in addition to stabilizing the protein, just those two proline substitutions increased expression of the MERS spike by about 50-fold over the wild type version. So both for preservation of neutralization sensitive epitopes and for expression from a gene-based vector, this creates some advantage. And fortunately, these two prolines were used in at least a dozen different coronaviruses over those couple of years, and it worked in all of them. Even some of the alpha coronaviruses were stabilized by this proline substitution between the helical domains of the fusion machine. So when this virus came, and we've been working with Moderna since 2017 on a pandemic preparedness concept we call prototype pathogen approach for pandemic preparedness. We've been using the MERS coronavirus for coronaviruses and Nipah for paramyxoviruses, and have made constructs and mRNA vaccines for both Nipah and MERS that had already worked and shown to be protective against lethal disease in animal models. And so because we had that 3-year relationship, and because we trusted each other, at least at the scientific level, we made an agreement with Moderna. Even though we planned to do the Nipah first, we said, we'll switch the demonstration project from paramyxoviruses to the coronavirus and use the new protein sequence here to solve structures and start vaccine development. Because for us, having the protein structure, knowing that it's in the right conformation, gives you the confidence to make diagnostics, to use it as probes to isolate new monoclonals like we did with Accelera, and then Lilly developed those into bamlanivimab and a new one we call it 1402. And then also apply that to vaccines. And not only was the structure and sequences useful for Moderna, but for several other companies. All the ones on this list except AstraZeneca used the two proline substitution in their initial vaccine antigen. So if you ask how you can learn from one vaccine to the next or one viral family to the next, it's pretty clear how RSV informed SARS coronavirus vaccine development, both from structural biology and protein engineering, but also for the use of monoclonal antibody therapy. And then I didn't talk about it, but there was a lot of concern over the vaccine-enhanced illness syndrome that was seen in animal models, the first SARS. And so all the work on RSV vaccine-enhanced disease from the vaccine in the 1960s was also relevant to getting this coronavirus started. And since the coronavirus vaccine program, not just at BRC, but around the world, has now made over 300 vaccine projects or concepts according to the WHO landscape analysis. It now has confirmed a proof of concept for this prototype pathogen approach for pandemic preparedness. It's shown the value of rapid platform manufacturing technologies, and it's introduced RNA as a real scalable, viable product to the world. And so together, all these technologies, and I haven't talked about the single cell sorting and analysis and the definition of antibody lineages that are all relevant to HIV, but together these new technologies make vaccine development more of an engineering exercise. We're not in the era of empiricism as Jenner and Pasteur started us out. We really can be more modular and more specific about timelines and specific goals. And these technologies can roughly be broken into things that improve precision of antigen design, either the target for antibody induction or the actual surface contours and structure of the antigen. And engineering these nanoparticles has really also increased the potency of these things. And then these other technologies can increase the speed. So for RNA, it was sort of it wasn't expected to the extent, I was a little surprised how unexpected the RNA efficacy was, but it was relatively unexpected. And we used it because it was rapid, because we showed with Moderna in 2016 that it was much more potent than DNA for Zika. We showed in the monkey model systems that we had, and it is even more rapid for manufacturing the DNA. So it is a platform technology. It's chemical synthesis, so it moves us away from the big campuses of bioreactors that have been needed in the past. It not only induces antibody, but also CD8 T cells and promotes the Th1 and TFH induction. We now have data in hundreds of millions of people that suggests it can be safe and efficacious. Unlike ad vectors and a few of the other vectors, there's no anti-vector immunity. The stability is being solved and the supply chain is improving. I think eventually this will be a lyophilized product that can be stored at room temperature. One of the things that I think is important is that this is a small footprint, small batch type manufacturing process, which makes it well suited for deployment into low and middle income countries. And not only would that improve the capacity for responding to pandemics and things like this, but it allows those local investigators to deal with their own regional diseases like Lassa in Nigeria or Rift Valley fever in Kenya, myorovirus in Mexico, et cetera. These are things that are pandemic threats, but really don't have a lot of commercial value or interest. But with this kind of rapid iterative design cycle capacity and relatively simple manufacturing, I think local investigators could start addressing these regional problems before they become global problems. And there is a lot of room for improvement in a number of different areas in codon selection, the secondary RNA structure. We don't really understand very well in terms of how it affects reactogenicity or translation efficiency or immunogenicity. And so this is important. And but finally, mRNA is not magic. If you don't deliver the right antigen, it's just not going to work. And we can go through more of the types of RNA that are out there. There's unmodified or modified nucleotides or self-amplifying RNA. The modified nucleotide that you've heard a lot about from Drew Weissman and Caitlin Rico's work uses 1-methyluridines or pseudouridines, avoids a lot of this signaling through toll-like receptors and intracytoplasmic receptors and diminishes a lot of the initial inflammation from a double-stranded RNA or a single-stranded RNA going into a cell. And it allows longer protein expression. And so it really has made a big difference in how well the mRNA is working. The problem is for HIV, I think we still have a lot of breakthrough questions that have to be solved before we can really get on top of antigenic variation, both on the B cell and the T cell front, the immunodominance problem, being able to target the right antibody lineages and make antibodies with the right physical and chemical features. And a lot of these problems exist also for improving our influenza vaccines. I think we have a lot to learn about maintenance of B and T cell memory phenotypes and inducing immunity in the right place. I think there's still a lot to do, obviously, on how viruses are evading immunity to begin with. And for viruses that, for HIV, for instance, where there's really never been a complete cure, self-cure without the benefit of CCR5 mutations or bone marrow transplants. And so there's just a lot still to learn and have breakthrough understanding to get to an HIV vaccine. I've envisioned how maybe some of these things that HIV vaccine development processes have promoted, like the conformational evasion, the structure-based approach that seemed to solve RSV, and some of the work we're doing on approaching genetic and antigenic diversity and immunodominance in flu. Coronaviruses have a lot of carbohydrate, not quite the glycan shield of HIV, but there's 22 to 25 glycans per protemer, so there is a lot of glycan there. And so one of the questions I've had is, can we take these different learnings from different viruses that are all made possible by HIV work and take them back to HIV and make more progress? The other thing I just want to point out, and some of you will appreciate this more than others, the ones who've been in this for so long, how, we've kind of fluctuated between a total antibody focus, then when we learned about clinical isolates and some of the problems of neutralization of primary isolates, we kind of went to a full CD8 T cell approach. And when that failed, we've gone almost all the way back to a full antibody approach. And I think unless we use both antibody and T cell effectors, we are not going to have a good chance to make an HIV vaccine. So in conclusion, before we go to questions, these new technologies have transformed vaccinology and are providing a lot of new options. And I think this idea of combining atomic level antigen design with nanoparticles and platform manufacturing provides a more engineering-like approach for vaccine development that may create even more solutions. But for HIV vaccines, I don't see anything in the near future without a few more breakthroughs. And so I just want to thank the people I've worked with over the years in the programs and the PIs at the Vaccine Research Center. That was a real privilege for me to work there, and my lab, and all the academic collaborators that have been involved in the RSV work. This is Jason McClellan, who's now at UT Austin. Neil King's been very involved in self-assembly nanoparticle component of our universal flu program, and all of our academic and industry collaborators that were involved in the coronavirus work and other extramural divisions of NIAID that were also very involved. So I will stop there, and we can talk about my pessimism or my reluctance to say that we're ready to tackle HIV vaccines.
Thank you so much, Dr. Graham. This is Monica Gandhi, and Mark Ansel and I will be moderating this. We actually have a number of questions in the chat. I'll just start with one that I really, I know HIV is going to come up very largely for this audience because the CFAR audience is but I just wanted to ask you about the advocacy that you have personally done for the Moderna vaccine and global vaccine equity. I know that there's this tremendous, that we haven't, yet hasn't acknowledged the contribution of you and the NIH, I think to the extent that is needed, but do you believe that the advocacy work that you and other academicians are doing are going to help expand the technology for global vaccine equity to the world?
Well, I see it as a big problem to solve. I mean, if you look at what's happened during this pandemic, the real problems we have right now are the emergence of variants. That is the key problem that we have. And the reason we have variants is because it's taking us 6 years to immunize the world instead of 6 months.
Right.
And until we can learn how to immunize the world in 6 months, then we're going to continue to suffer from this problem, even in future pandemics. And so, to me, the two big problems there are, one, vaccine hesitancy by people who have access to the vaccine. And the other problem is lack of access to the vaccine, largely in low and middle income countries. So if it takes us this long to even get more than 10% vaccination coverage in Africa, that is where all of our variants are going to come from that put our original products at risk of failure. And so if we can't solve those two problems, then I think we haven't made very much progress towards being prepared for the next pandemic. And so a big part of the low and middle income country improving their capacity, to me, is involving something I mentioned already, which is moving manufacturing capacity and research capacity into places where most future pandemic threats will emerge. Most pandemic threats that are not commercially viable targets. And so I really think we need an entirely new paradigm for how we think of making products for public health. And you can't really expect a company to solve all those problems. I think we need nonprofit and government and philanthropic and government to government kinds of collaborations to solve some of this. And I think mRNA gives us a big opportunity for that. And so to me, the hope that mRNA represents is if you can do chemical synthesis in a small modular containment facility almost anywhere on Earth, and then make at a cost-effective way tens of thousands of doses instead of hundreds of millions of doses, then you can start solving some of these regional problems. Like, why is Ross River virus, an alpha virus in Southern Australia, still infecting 50,000 people a year? It's because it's not a commercially viable target. So even in this high-income country, you're not addressing these kinds of problems that could become pandemic threats. And so, I don't really, I don't understand business or the company models well enough to say very much about that, but I do know we need a different paradigm for solving some of these problems. And if you had this kind of rapid iterative design cycle type of technology in Kenya or South Africa or Rwanda or Senegal, and they were able to support their investigators solving their regional problems, they would then also be the surge capacity that we would need during the next pandemic. Because one of the problems is, it's hard to be totally ready for this kind of surge capacity. And we need more distributed manufacturing, in my opinion, to be able to do that. So that was a long answer. I may not have answered your question, but
you did, and thank you for your advocacy. Yeah. And I'll let turn it over to Mark for more questions. Thank you.
Yeah, that's really related in a way to the question I most wanted to ask. And I want to say again, thanks for being with us and for the great presentation. It really spoke to the answer to the question I think a lot of people in the public, but even those of us who are pretty deeply involved in this kind of research had at the beginning, is how did they do that so quickly? And it really comes down to preparedness and technologies that put people in a position to be able to solve this problem. So the question I wanted to ask which is a little related to what you were just talking about, it maybe beyond the social and government policy factors. What are we lacking to really accelerate the extensibility of this technology? Are there also scientific or technology shortcomings that could help us to really enable the kind of things that you were just discussing about being able to rapidly respond to regional threats? And I know you've spoken to the specific problems with HIV, but even outside of HIV, this holds enormous promise. And what do we need to do to really realize that promise?
Well, RNA does have potential for solving some problems and even maybe replacing some older vaccines that now have fairly complicated technology and do take a campus of bioreactors to actually make. And so the problem is if you're talking about doing something in the next 2 or 3 years, it's pretty much going to have to be paternalistic, giving Western products into Southern Hemisphere countries. But if you talk about a 20-year plan, like there has been going on in India for the last 30 years, 30 years ago, India couldn't make vaccines either. And now they've developed the technology for biosimilars, for monoclonal antibodies. They make almost half of the vaccines that are used in childhood campaigns around the world. They can make their own vaccines. And it's been because of a 30-year investment in generating the technology infrastructure. And the problems in many parts of Africa are at very, very basic levels. Like I always bring this up, but who's going to calibrate your micropipettes? We need technical people who can service equipment, who understand things at different levels. You have to have somebody who repairs the flow cytometer. You have to have somebody who does the supply chain and makes nucleotides and lipids and syringes and glass vials and rubber stoppers. And so you need all these things, technical things, and a lot of technically trained people to support the work. And I think if you don't start at that basic level of building the infrastructure that would then support the manufacturing, or at least research level grade manufacturing, I just don't think it's going to have long-term success. And so far, I don't think people have seen this as a good enough investment, or they're reluctant to make that kind of investment. But if we've really had 13 or 14 trillion dollars pulled out of the economy, then how many technicians could you train and how many laboratories could you set up for $13 trillion? And I think that the time might be right, the technologies might be right at this point in time to try to convince people that this kind of long-term investment should be made to develop the infrastructure, to put the people and the facilities in place that could do this kind of work, to improve the reliability of the utilities of water and electricity. And all these things really just need some basic support.
I have an audience question specifically asking about HCV mRNA vaccine development, and I wonder if you could also expand there on what the particular challenges are with you talked about the importance of antigen design and picking the right targets. What are apart from HCV, what are the low-hanging fruit, and what's the risk reward for deploying this for iterative design for variants, for other viruses? What's both the limiting factors and also the real easy opportunities?
Well, HCV and maybe some other flaviviruses and influenza and HIV to me are the really, really big, difficult problems. And there are groups Pamela Borkman and Justin Bailey at Johns Hopkins have done some really beautiful structure-based design work with some of the HCV antigens. Those could probably be delivered by mRNA. The big advantage you have in HCV is that about half of people can cure themselves. So there may be really some chance of fully aborting the infection if you have a good enough CD8 T cell response or respond quickly enough to the infection. So there is a lot of antigenic variability that is going to be a challenge, but I think there's at least a better chance there to have a vaccine if you can activate the right effectors and have a little bit of antibody at the beginning that can knock down the inoculum side. So the T cells have a better chance of clearing the virus infected cells. So I think this structure-based design and maybe display on particles and maybe delivering by gene-based vectors or mRNA would have a good chance at HCV.
Thanks. I see a hand. I'll let Steve Beeks ask this question himself if you come on mic, Steve.
Hey, Barney. So I really appreciate your global perspective here, this big picture discussion that we're having. And I was wondering if you could talk a little bit about where you think the world is going in RNA vaccines in two issues. One is germline targeting for prevention, and one is the role of RNA vaccines in therapeutics for HIV for cure agenda. Those are two big issues, but I would love your perspective on the challenges and promises there. Right.
Well, what used to be the biggest problem was knowing if you could even scale it up. And now we know that we can scale it up. And you can generate a supply chain of nucleotides and lipids and actually do this on a large scale, which eventually will make it less expensive and maybe more feasible to use as a therapeutic approach. But how you would use it as a therapy, you can express monoclonal antibodies from RNA, especially if you give them intravenously. But I think a lot of that safety and kind of clinical work, getting that to really work on a long-term basis with long half-life antibodies and things, I think is still a fairly long way away. If antibodies could be therapeutic, I'm not sure that they can. If mRNA could deliver things to a particular cell that could, liberate the virus from that latently infected cell, then I don't know what that compound would be, but if that could be done, I can see how that could maybe at least expose the latent virus and reservoirs to being cured. Delivering things as either particles or specific antigens that would target a particular antibody germline that would have capacity for making broadly neutralizing antibodies. I think for a vaccine, I think there's some progress there, but I think we have a long way to go. You probably need to target more than one germline, and probably, if you had a half a dozen germlines like we do for influenza that we know can become broadly neutralizing antibodies, I think we need more of those germlines for HIV that would be more reliable. And like I was saying, I don't really think we're going to get this just by antibody alone. And I think antibody can reduce the inoculum size and give you a much better chance at clearing infection or controlling infection before you have these high virus loads. But I really think that you need to have T cells present that can control things early on. And if antibody can keep it regional and T cells can clear it in 14 to 28 days before it gets into lymph nodes, then maybe there would be a chance to do that. But I think just targeting germline B cells, germlines is, I don't see that as enough to really have a fully preventive or controlling vaccine. I don't know if you, I'd like to hear comments, because I don't want to just say things that are pessimistic, and I'd like to hear somebody who thinks has a little bit more hope about this.
Yeah, you are a bit pessimistic for someone who basically just had a miraculous 2 years with COVID. I would think you'd be more positive about HIV. And
well, I've always felt about HIV that I wasn't sure if it was possible, but I think it's imperative that we keep trying to make an HIV vaccine. And because I think trying to make an HIV vaccine will basically solve all the other problems that we need to solve just about every other viral disease. And if we can really figure all those things out and maybe then apply the individual virus findings or successes back to HIV, then maybe we can even make an HIV vaccine. I really am, I'm optimistic, but more in the long term. And I really think it's critical that we keep working on an HIV vaccine, because solving all these problems is what is going to solve everything else.
Some other political questions, Mark, I'll turn to one more, which is around the vaccine hesitancy, actually, that someone's asking in the chat. So, what I think we all understand that this was novel, and so it has led to less vaccine confidence. But have you been astounded by the degree of misinformation and lies? I guess from a governmental level, what is there any? I mean, can you be on TV every day? Like, what is it that what do you think has contributed to this degree of misinformation in the current era? I know it's a complicated question, but it's being asked, and it's a fair one.
I'm not sure if being on TV every day improves or decreases your credibility. That's one of the problems that we have. I am disappointed, but I'm not that surprised about what's happened, because there's always been about a third of people who don't trust institutions, who don't trust governments telling them what to do, who don't trust expertise. They don't trust the way that message is delivered. And so I think we just have to do a lot more work in between crises. When we're not in the middle of a crisis, I think we have to do a lot more work in education of biology and helping people to be more prepared for things that come in the future. The problem of working in crisis is that you just don't have time to build trust in that way, and so there just has to be a different level of trust, and that is a long-term process. And it's what's really become more clear to me is that despite these really amazing engineering and biology technologies that have been evolved, our infrastructure, the way our social networking works, the way our politics works, the way our moral compass works, none of those has advanced the way technology has advanced. And it's like we don't know what to do with the technology, because we're still operating as if it's 1918, because the politics and the social structure and the moral basis for how you do things is about the same as it was 100 years ago, even though the technologies have all changed. And so now with, we have now this social media and, mass communication, but we don't have any kind of rules for how to do this. after newspapers were invented, then there was a whole series of journalistic ethics things that were established and kind of rules of engagement for what is journalistic integrity. And I just don't think we've evolved socially well enough to know how to manage social media. We don't really know what we're doing. We're just throwing things out there, and so we need social scientists and politicians to kind of evolve the way some of the technologies have evolved. And I don't see a lot of that happening yet. I'm hopeful that it will because it's so needed.
Monica, I think that you're muted or your mic.
Oh, sorry. I was just going to say there's going to be a lot to write about. I'll let you Steve asked the briefest question because we want to know, he wants to know how fixing HIV will fix everything else. We love that on the CFAR comment. And then I'll let Mark finish with last questions.
Well, I think if we really could understand how to deal with antigenic variation, then that would help us in flu and HCV. If we could really learn how to deal with immunodominance and avoid targeting things that are not helpful in terms of immunity, then that would also help us with influenza especially. And if we could learn how to target specific germlines of B cells, that would help us with things like influenza. If we learn how to avoid latency in CD4 T cells, if we learn how to avoid sequestration into immunoprivileged sites, if we learn how to, all these things that HIV can do, if we learn how to deal with glycan shields and figure out ways of making antibodies that have exactly the right angle and exactly the right chemistry with maybe a little negative charge at the tip of a long CDR3. I mean, if we learn how to do some of those things, I think it just makes it possible to create immunity to a lot of other problems.
It makes sense. Thank you. I'll let you finish.
Well, I have unfortunately too many questions from the chat to ask them off. I think I'm going to close up with one that I prepared before, and it relates to what you were just talking about. You spoke about pandemic preparedness, kind of becoming a catchphrase, and you talked about it in the context of your collaboration with Moderna on prototype pathogen approach. And this discussion about HIV also sort of brings up what we can do to have an impact when these kinds of global health challenges arise. So is there some wisdom you can share for so many of us who pivoted and tried to do what we could in the moment for the future, from a research and development perspective, maybe? What are your thoughts about how we should structure our institutions to be poised to have an impact for challenges that may arise in the future?
One thing I think that could be done is for academic institutions to find partners in low and middle income countries to support the infrastructure, because I don't think we can count on commercial entities to transfer technology. And, it'll be 3 generations behind if they do. That there has to be a different way of developing technology, I think. And so, if you have an engineering school that knows how to do GMP, then why don't you choose a country like Senegal and help that country learn how to do GMP work? And if you have structural biology, maybe you can partner with them until they have enough resources to have their own equipment. But if you had structural biology in one site and mRNA manufacturing or research trade, at least in a low and middle income country site, you could help design antigens and they could be involved and make the product, do the testing, and get the animal studies done and maybe support the development. And I think that mRNA creates all these options because it is relatively simple chemical synthesis. It's not that simple, but it's relatively simple compared to bioreactors. And if you can do chemical synthesis and have ideas and know what your local targets are, you could make a lot of progress as this infrastructure is being built. So if governments and philanthropists can build the infrastructure and academic groups can support the knowledge gains, RNA is also simple enough, I think, that and new enough that I think that there's a lot more that can be discovered. If you can unlock how to predict how secondary RNA structures is going to affect toxicity and expression, then you could license that technology and the companies over here would have to buy it back. So I really think there's opportunities like that where there could be role reversal, where there have to be licensing back to high-income countries if this kind of thing was set up in low-income countries.
Well, I want to acknowledge the importance of your open communication about the stabilizing mutations for the spike too, and how that accelerated things. So you put your money where your mouth is with that philosophy. So thank you again.
It's one of the advantages of being in government. So it allows you to do things like that.
Thank you so much, Dr. This was one of our best attended CFAR seminars. We're incredibly honored to have you come and speak about your pivotal role in developing this one of the most important vaccines of the century and a huge advance in technology. So thank you very much for coming on behalf of the CFAR and Mark ImmunoX. And thank you again for coming. We really appreciate it.
Nice to see you all.
Thank you.